Medicenna is a clinical stage immunotherapy company focused on developing novel, highly selective versions of IL-2, 4, and 13 superkines, as well as first-in-class empowered superkines. The company has just presented updated clinical data from the ongoing phase 1/2 Ability 1 study evaluating MDNA in patients with advanced solid tumors. I've got CEO Fahar Merchant here to help us review and understand that data. I'm Martin Gagel with Market Radius Research. Please remember this is neither a recommendation nor investment advice. We're here to learn about the company. Fahar, thanks for joining us.
Thank you for having me. Really appreciate it.
Before we get into the data itself, well, I think we should step back a bit. And can you just explain in simple terms, this is a phase 1/2 study, and what were you trying to accomplish?
Yes, so let me go a bit, provide you a bit of background around the IL-2 space in general. As you know, and the audience probably is aware that IL-2 has been around for the past 30 years and has shown in a small percentage of patients durability, long-term cures, etc., in patients with melanoma or skin cancer and also kidney cancer. The problem has always been that it is incredibly toxic in the version that was first commercialized. And that has been the big issue about having a safe IL-2. So, one of the key purposes of this particular clinical trial is really to establish whether the engineering we've done on this molecule, the way we've designed this molecule, is it safe? Is it able to stay in the bloodstream long enough so the patients don't need to get a treatment 3 times a day, which is the case with the old version of the IL-2? So what we are trying to study is how can we make this drug safer, easier for the patient so that they don't have to be admitted into intensive care to receive the drug because the old version had potentially life-threatening side effects. Third is from a patient's convenience so that they are not receiving this drug 3 times a day for 5 days and then repeating the process 2 weeks later again. So trying to see, for instance, has the design of the drug able to accomplish our objective of being able to treat patients once every 2 weeks or once every 3 weeks in an outpatient setting, and therefore achieving that key objective of safety and the ease of administration. But that's not all. We wanted to also see,
Fahar, can
yes,
I pause you here for a sec? Before we get into the other part, on that initial part of what you were trying to accomplish, how did the data show against those milestones or those goals you were trying to do?
and this is exactly what, we are delighted with that, the results from a safety perspective. We were able to demonstrate that the drug is relatively safe, that can be administered every 2 weeks or once every 3 weeks, either on its own or in combination with another drug, which is Keytruda. So, all in all, from a monotherapy, meaning MDNA11 on its own, or in combination with a checkpoint inhibitor, we were able to demonstrate the safety of this drug and the ease of administration for the patients in patients with late stage disease who normally would be less inclined to tolerate such a regimen of treatment. So that's basically was accomplished.
You got the big checkbox, but then also while you were doing this, you were looking at some efficacy measures as well on a variety of tumors.
Yes.
And so tell us about what you saw there.
Yes, yes. So, that was one of the key objectives. Having tested so many different tumor types, we were able to first and foremost demonstrate that the drug has a really strong uh, or really solid outcomes or response rates in patients with melanoma. So these patients were all previous, had failed standard of care, including checkpoint inhibitors. And we're seeing response rates in these patients of about 38%, which is way above the benchmark of 30% that is currently as a threshold for getting accelerated approval, for instance, particularly in checkpoint failed patients. So that's 38% is very impressive. The other thing that I think, just as a side note here to add, is that it's important to note that every drug to date that has been approved for melanoma has subsequently been approved for other solid tumors. So, in a sense, here we are showing that this drug is being pursued and the data looks really solid with melanoma. And as a result of this solid data, you also heard last week that we had started enrolling patients in a new clinical trial funded entirely by the Melanoma Foundation in Italy in patients with melanoma prior to surgery. So these are very early stage patients who have not even had surgery, have not received any prior therapy. And these patients will be part of a study which will be comparing MDNA11 with other approved therapies. So that is a strong, important milestone. And we believe MDNA11 could be really groundbreaking in these early stage patients. Plus, we've already seen that the drug works in nearly 40% of the patients who failed a checkpoint inhibitor. So combined, melanoma obviously is a very attractive target or indication for us to pursue. The second one where we saw activity was also in patients with a biomarker known as MSI high. And both of those patients that were MSI high turned out to be patients with late end stage pancreatic cancer, and they had both failed a checkpoint inhibitor. So that, as you know, pancreatic cancer is a tough disease to treat. It shows that MDNA11, in addition to melanoma, has a solid impact on patients with other tumors like pancreatic cancer. In combination, we found that patients with endometrial cancer, so this is a gynecological cancer where there has been no prior history of IL-2 showing any activity in this particular tumor type. And here we see for the first time our IL-2 with Keytruda having patients respond with endometrial cancer who have previously already failed multiple therapies, including Keytruda. So these patients, we're seeing a response rate of 50%. I know it's a few patients, it's 4 patients, but all of these 4 patients had failed therapy. One of them had a stable disease. So here you're looking at disease control rate in end-stage endometrial cancer of 75%, which is really exciting. And then also, we found patients with a different biomarker called TMB high. And here we saw patients with colorectal cancer, with breast cancer, and also with anal cancer. These patients had responses to this drug as well, where typically these patients who have failed a checkpoint inhibitor, repeating these patients or treating these patients again with a checkpoint inhibitor does not generally result in favorable outcomes for these patients. So we are seeing multiple tumor types, but by and large, I can sort of group them into 4 tumor types, at least for now, that looks really promising. Melanoma, uh, MSI high tumors, particularly pancreatic cancer patients, uh, endometrial cancer patients, and also patients with a biomarker known as TMB high, where we've seen responses in patients with colorectal cancer, bladder cancer, and breast cancer. So that's really exciting for us to see multiple, at least 4 different indications that we can pursue.
This was not the final data readout from this study. This we're getting more, we will be getting more data in 2026, correct?
Correct, yes. And just to let you know that the data readout was based on the response data was based on patients that had at least one scan post-treatment. So it is really important to know that although, for instance, if you look on the safety number of patients that were evaluated for safety, those were 61 patients. So any patient, irrespective whether they got a scan or did not receive a scan yet, those will be counted all in the safety portion of the study. And then, of course, there is a group of patients we call efficacy evaluable, meaning those were 55 patients. And those 55 patients out of the 61 had at least one scan. Okay, so that's the patient population that we know whether the patient had PR, stable disease, or progression. So those are the numbers that we need to use. And then finally, out of the 55, we had 7 patients that were part of what we call dose escalation. We were testing the drug at incredibly low doses. When you start a clinical trial, particularly first in human, where you've never tested this drug in a human before, you try really low doses of 3, 10, 30 micrograms. This is where the drug was initially tested. And therefore, we know that those doses did not result in any meaningful responses in these patients. And when you look at the dosing at which the drug was active, which is 60 micrograms and above, that is 48 patients. So the denominator in any calculation for response rates in single agent therapy should be 48, not 61, which has been used by one of the analysts.
Is that N of 61 the patients? Is that how many is going to be the total, or are you adding any more to that? And then when should investors expect to get another data readout in 2026?
Right. So the current protocol, the way it's written, it says that we are able to treat up to 15 patients per tumor type. So we have 4 tumor types. So that would be 60 patients in the monotherapy and 60 patients in the combination. Okay.
Okay.
Now we've already treated about 110. So really speaking, there is another 10 patients that we need to enroll and treat. But remember, a number of those patients were with other different tumor types. So potentially looking at a dozen or 2 dozen more patients. We're expecting that we would enroll all of these patients by the end of this next coming quarter and have
The end of Q4 or the
oh,
end of
uh,
Q1?
Q1.
Okay.
Yes. Q1 will finish enrolling all these patients, possibly earlier. And the key thing here is that we need to make sure that we get at least one scan from these patients.
Yeah.
And the data, as it is accumulating, will allow us to meet with the regulators. And the plan would be to meet with regulators in late Q1 or Q2 of this coming year to determine what the pathway is for this drug. Clearly, right now, we are seeing for these 4 different tumor types, melanoma clearly is way ahead and becomes potentially the first indication that we'll be pursuing or engaging the regulators with respect to MDNA11. Which one is going to be the second or third one? I cannot say until we have more patients with endometrial cancer, MSI high, and TMPI tumors as well. So those will mature as those data mature for those patients, and we have some additional patients with earlier stage disease. We'll be able to determine which other tumor types we should be pursuing with the FDA. Okay.
Okay. And would we get some public data readout in Q1, or would that maybe be in Q2, or what is your sense on that?
I think it'll probably be in Q2. That's more likely in
Okay.
Q2. In Q1, though, just to give you a bit of background, is we have a bispecific IL-2 anti-PD-1 molecule. It's called MDNA113. That was the subject of a large $11 billion transaction between Takeda and Innovent, a Chinese company. We think we have a much better drug than Takeda's new drug. And this particular drug, we're currently not in the clinic, but we will be presenting non-human primate data sometime in January. So we are conducting a study to demonstrate safety of this drug, MDNA113, in monkeys, and that will help us establish First and foremost, whether the drug is safe before we start the human clinical trial, which is likely to commence in the second half of next year.
I'd like to talk to you about how your share price has responded to last week's news. There's often the old adage, buy on rumors, sell on news.
Yes.
Um, the data, to me at least, seems quite positive on there. To be honest, it's a little confusing. There was a lot of different data. It wasn't clear cut. I suspect that people were maybe thinking this was more like a phase 3 trial where okay, do we have a hypothesis that we're testing and do we hit a benchmark? But it was more collecting a lot of signal. Um, any thoughts on why the share price responded the way it did on what appears to be solid, good news?
Yeah, I think, there is obviously volume associated with news like that, and some investors may find this as an opportunity for profit taking, who might have bought the stock at, as you recall, a year or two ago, we were at 20 cents and so on and so forth. So it was a perfect opportunity for some of them to mostly profit taking, I would say. Others that may feel that we're expecting a lot more, as you say, potentially a data set that would be more akin to a statistically designed randomized clinical trial, which this certainly wasn't. And the sort of perhaps the mixed messages that went out there from potentially analyst reports and so on and so forth. So I think the key thing here is to understand that there could be many causes or many reasons, but the data is solid. So that goes to show that this drug is so much better than the original drug and so much safer and so much more efficacious. So we're excited. I mean, that's, suffice to say, we are very pleased with the data so far. We think that once the message gets cleared up, I think there will be interest again in the story. I think we are really confident that as the data matures and as we enroll patients with earlier stage tumors, we'll see that as we enroll patients in the neoadjuvant study, the NeoSite clinical trial in Italy, that will add additional, support to the story. We've seen KOLs during our webcast last week who were really excited with the data that we shared with them. They are world-renowned clinicians in melanoma, in kidney cancer, in other tumor types, etc., who were surprised to see how safe, but at the same time, how effective this drug was. And to quote them, they called it the best-in-class IL-2. So really excited about that.
Fahar, that was great. Thank you. And we're coming up to 2026 here already. So we've talked a little, some of the data points and expectations for 2026. Can you just summarize what are the key milestones or expectations investors should be keeping their eyes out for in 2026 for Medicenna?
Well, multiple, really. The first one that I mentioned earlier was right out of the gates in 2026. We'll have some non-human primate data on MDNA113, so that be the first data set that we'll share. We will obviously have, as you can imagine, more data from more patients in the phase 1/2 study, including the patients that have not received their first scan, for instance. So we expect that late Q1, early Q2. Again, this will be for both the monotherapy and the combination arm of the clinical trial. We'll have clarity with respect to our next steps with respect to the melanoma indication, particularly with respect to what our plans are for a potential phase 2 registration trial, our interactions with the FDA, etc. That should be coming in Q2 of next year. We will also have early data sets from the NeoSite clinical trial, which is the neoadjuvant clinical trial that's been conducted. Here, the important thing is that patients really get their tumor removed 8 weeks after treatment with our drug. So in a sense, within 8 weeks, you know whether the patient had a partial or a complete response relatively quickly. So that's another set of data that we'll have adding on. In the second quarter, we'll also have clarity as to what other indication beyond melanoma. Is it going to be MSI high, TMB high, endometrial cancer? Is that a second indication that we'll be pursuing? Of course, we'll be looking for more data there. So that will add to the story in terms of, how do we grow the MDNA11 franchise beyond melanoma into other tumor types where IL-2 has never been tested before. So that's the data set. And then in the second half of next year, of course, we will have ourselves filing an IND for MDNA113, which is the bispecific anti-PD-1 IL-2 program, and have early readouts on the first patient population that gets treated in a phase 1 setting with MDNA113. So we will have a really solid guidance in terms of not only melanoma, but other tumor types with respect to MDNA11, what are the registration pathways, the fuller data set, more mature, including multiple scans from these patients. But finally, of course, a clear guidance from the regulators as to which indications, in addition to melanoma, we should be able to pursue.
Thank you very much for that update on the data and some outlook into numerous data points coming out and some substantial ones as well coming out in 2026. So thank you very much for the update.
Thank you.